ऑल राइट, सो आज हम डिस्कस करने वाले हैं नेचुरल किलर सेल्स के बारे में। नेचुरल किलर सेल्स एक तरह से लिम्फोसाइट्स के टाइप होते हैं। ऑल द लिम्फोसाइट्स आर वाइट ब्लड सेल्स एंड दे टुगेदर, lymphocytes are B lymphocytes, T lymphocytes and natural killer cells, that together constitute 20 to 40% of white blood cells. So you can say these are a kind of white blood cells. All right. If you turn the page, you will find a table, right? So this table is showing the differences between innate and adaptive immune responses, page number 499 in your Pathfinder. If you look at the differences, please, I want to show you this one. So the major cell types in innate immune response, if you see, these are phagocytes. We already have covered major phagocytes, which are neutrophils and macrophages. Eosinophil also perform this phagocytosis, but generally they kill the parasites like helminths or the worms, which are multicellular organism, right? And the bacteria and other entities which are foreign to the body are killed by phagocytes. Generally, the first cells which reach at the site of infection are neutrophils, but the macrophages are also very efficient phagocytes because of their large size. और लास्ट क्लास में मैंने एक चीज और बताई थी कि ये अगर मान लो कोई बहुत बड़ा फॉरेन पार्टिकल है जो एक फैगोसाइट या एक मैक्रोफाज उसे किल नहीं कर सकती, so macrophages also having this property to fuse together to make a giant cell. तो वो एक बड़ी सेल बना सकते हैं और फिर फैगोसाइटोसिस को परफॉर्म कर सकते हैं after fusing so many macrophages, right? और भी बहुत सारे डिफरेंसेस हमने लास्ट क्लासेस में कवर करे हैं। तो आज हम डिस्कस करेंगे नेचुरल these cells, these are called NK cells or natural killer cells, which are actually a type of lymphocyte, because we know there are three types of lymphocytes: B cells, T cells, and third is these natural killer cells. But these B and T cell will make the adaptive immune response. These are major cell types which we will also discuss in our adaptive immune response, right? But first, because we are going to finish this innate immune responses, we need to cover this portion called natural killer cells. Now see this, from where these cells are coming and what is the property of these cells and how they are similar to the other lymphocytes. What are the major differences? Why we are placing these natural killer cells in this group, not in this group, whereas the other lymphocytes are major type of adaptive immune response cells, right? But natural killer cells are different because they do not have any antigen recognition receptors over their surface. Therefore, we have to place them in innate immune response, right? Whereas the B and T cells do have the antigen recognition receptors over their surfaces. Therefore, we will place them into the adaptive immune response. So quickly, we will go through through these differences, and then we will see how these natural killer cells are formed and what are their major roles. So if we talk about in the terms of what are the major properties, characteristics of the adaptive immunity. So adaptive immunity will have an antigenic specificity. This is very much specific to the antigen or the challenge or the foreign particle which is coming from outside in the host body. So adaptive immunity, also known as specific and acquired immunity. Now, please understand this term, specific. It is very, very much specific because adaptive immune response is due to the presence of some membrane receptors over some specific cells like B cells and T lymphocytes, right? So it is also called acquired one. Why acquired? As we have discussed the innate immune response. So innate immune response is a type of immune response which is present in an organism since its birth. लेकिन बर्थ के बाद में जो आता है उसको बोलते हैं एक्वायर्ड, यानी कि जो हमने बर्थ के बाद अचीव करा है, यानी कि जो हमारे एक्सपीरियंसेस हैं हमारे इम्यून सिस्टम के, हमारी बॉडी के, जो चैलेंजेस उसने फेस करे हैं, जो अलग अलग टाइप के इन्फेक्शियस पार्टिकल्स को उसने रिमूव करा है. So those acquired immunity is actually called the adaptive immunity, right? It is capable of recognizing and selectively eliminating the specific foreign antigens. So it is very much specific and it can recognize a vast array of antigenic challenges or epitopes which are present over the antigen. These are specific molecules. Epitopes also we have covered in class. So epitope will bind with the paratope region of the antibody, and in the antibodies, the regions which will specifically bind with the epitope region of antigen are known as complementary determining regions, CDRs, right? Or the variable regions, which are of three types: CDR1 to 3, and CDR3 are the major variable regions. So they are showing the maximum diversity. All right. So these all things, if you go into the previous lectures, you will find everything we have discussed. Now, the second property of adaptive immunity, because it is very specific and it will recognize, capable of recognize a vast array of foreign challenges, uh, antigenic challenges. So it does not come into the play until and unless there is an antigenic challenge. इसको acquired इसलिए ही बोलते हैं क्योंकि ये तब तक काम नहीं करता जब तक कि कोई antigenic response, मतलब एक तरह से challenge या कोई infectious particle body को encounter नहीं करता. So it does not come into the play until there is an antigenic challenge to the host organism. So following characteristics. So four major characteristics of this adaptive immunity are antigenic specificity. Very, very important is immunogenic memory, which is actually not shown by the innate immune response. And many times the question has been asked from this one. So memory is one type of characteristic which is shown by this adaptive immune response, and it is absent from the other type, right? So it is ability to recall the previous contact. इसको बोलते हैं secondary immune response. So there are two type of immune responses depending upon whether a foreign particle or a pathogen is infecting first time, or it is infecting second time, or after second time, right? So if our body has already encountered this pathogen previously, known as primary response, so जब वो दोबारा infection करेगा या contact में आएगा हमारे immune system की, our immune system will have some kind of memory response. Memory cells are formed and stored. So these memory cells will recognize that particular pathogen very efficiently, and it will mount a very severe immune response very quickly. So there are two things. One is time restriction, so it will take less time. Second is efficiency. Efficient immune response in the sense that our body will make large number of cells will be activated simultaneously. So it is a acute response in the sense. So ability to recall the previous contact with the foreign molecule and respond to it in a learned manner. So this term is also very important. In a learned manner means our immune system already learned about the pathogen or the infectious particle. That is with a more rapid and large response. So there are two terms I told you. One is it is rapid, it will response is fast, and the response is large in the sense the large number of molecules and cells are formed and activated in this particular response. Diversity, adaptive immune response showing a large diversity in comparison to great diversity in comparison to innate immune response, right? So ability to respond to different epitope even if the individual have not previously encountered them. So this is the property of this adaptive immunity. So from where this characteristic is coming? So this is coming from the, because our B cells and T cells, which are having antigen specific receptors over the surfaces, they will have different specificities. So the diversity is already there, and they can recognize a large number of antigenic particles or the epitopes, even if they not able to encounter with any pathogen previously. So already they are having so much diversity. We will discuss this thing in great detail. So I will tell you some numbers so that you can get this term better. And the last one is self, non-self recognition, which is the ability to recognize and respond to the molecules that are foreign and to avoid making a response to those molecules that are self. So our immune system is always having the self, non-self recognition, whether it is adaptive immunity or innate immunity. It is self-tolerant. It is showing the self-tolerance. It is self-tolerant to the molecules which belong to the host body. And it will only mount an immune response to the pathogens or the foreign particles, which are actually not part of our body. All right. So this property is important because if it is not self-tolerant, it will mount an immune response against the body's antigens and it will harm the host somehow. Now, first of all, see this particular diagram, though we have discussed this pathway many times. So this is called hematopoiesis, page number 28 in your QA. So what it is showing that in the bone marrow, there is hematopoietic stem cells. Hematopoietic stem cells are kind of cells from where all the different types of blood cells are formed, whether they are myeloid cells or lymphoid cells, all are coming from the same hematopoietic stem cell, HSC, right? Now, there are two type of progenitor cell. One is myeloid progenitor, one is lymphoid progenitor, which are coming from this hematopoietic stem cell, which will differentiate into different cell types. So we have already covered this myeloid progenitor and myeloid population of cells, including the neutrophils, monocyte, macrophages, dendritic cell, eosinophil, basophils, platelets, and red blood cells, RBCs, erythrocytes, right? In the today's class, we are mainly focusing over this natural killer cells. So actually, these are coming from the lymphoid progenitor. You will see here, this hematopoietic stem cell will differentiate into a lymphoid progenitor cell, and it will differentiate finally into a natural killer cell. We will discuss the characteristics. Okay, so first of all, understand how these are formed. So the natural killer cells are coming from the lymphoid progenitor cell, which is actually coming from the hematopoietic stem cell. देखो hematopoietic stem cell तो किसी भी cell type में convert हो सकती है, either myeloid or lymphoid. Myeloid can give rise to different type of cells, but once they are making these myeloid and lymphoid progenitor cell, then their fate is decided. Either they will convert into the natural killer cells, or these lymphoid progenitor cell will give rise to the T and B cells, which will further differentiate into the T helper cells and T cytotoxic cells, these T cells, right? And the B cell, which is actually, this is called the naive B and T cells, it will differentiate into a large plasma cell, which will secrete large number of antibodies. So these two cells called B and T cells, they are part of adaptive immune response, as we already have covered. But this natural killer cell is actually not having, as you can see the cell, it is not having any surface receptors over this cell. Therefore, they are part of innate immune system, innate immune response, because they are not having any surface receptors for antigens. All right. So next we go to page number 32. All right, so 32. Now, we recently have discussed from the diagram that hematopoiesis is the process by which hematopoietic stem cells develop into the mature blood cells. And these mature blood cells are of different types: neutrophils, eosinophils, basophils, monocytes, and red blood cells, natural killer cells, B cells, T cells, all are white blood cells. And out of these white blood cells, we know the three different type of cells called B, T, and NK cells are constituting 20 to 40%. This we already have covered. Now see this hematopoietic stem cell that is induced to differentiate loses self-renewal capacity. Obviously, if it is having this self-renewal capacity because it is a stem cell, but if it is different, it is induced to differentiate undergoing hematopoiesis, it will lead to the synthesis of two types of cells: common myeloid erythroid progenitor cell, that is actually called CMP, or CLP, common lymphoid progenitor cell, which will have decided fates, and they cannot self-renew themselves because they are not stem cells, right? They will differentiate into different cell types, but they will not have this property. So this property is lost if the cell is induced to differentiate. All right. So we already have discussed that the common lymphoid progenitor, which give rise to the B lymphocyte, T lymphocyte, and natural killer cells. This we already covered. Now, okay, so first we will go to 37 now. All right. So now we directly come to the lymphocytes. So lymphocytes are the principal cell players in the adaptive immune response because when we are why this statement is written here, although we already have discussed that the lymphocytes are of three types: B cells, T cells, and natural killer cells. But this is a contradictory statement which is written in this line, that the principal cell players in the adaptive immune response, but we are discussing lymphocytes in innate immune response. All right. So the reason is actually there are three types of cells: B cells, T cells, and natural killer cells, right? Out of three, the two are actually present in the adaptive immune responses. Only natural killer cells are part of innate immune response. And you need to note it here that the natural killer cells are only 5 to 10% of all the, wait a minute, so I'm trying to write it here. So they are only 5 to 10% of all lymphocytes. Now please note it, lymphocytes. Why I am writing it? Because the natural killer cells are only 5 to 10% of lymphocytes. Now see the terminologies, there are differences. First we discussed that the natural killer cells are one of B, T, and NK. So they together are called as lymphocytes. And I told you that they together constitute 20 to 40% of leukocytes that are actually called WBCs, right? White blood cells. Now see the differences. These lymphocytes are actually 20 to 40% of white blood cells in the blood. But out of these lymphocytes, which are actually three different types of cells, only 5 to 10% cells are lymphocytes, are actually the natural killer cells. Now you can understand that rest of the lymphocytes are actually taking part in adaptive immune responses. And you can also get an idea that, okay, so if 5 to 10% of lymphocytes are making natural killer cells, we are left with 90 to 95% of cells. Yes, if you want to ask a query, please write in the chat box. So these cells are, or these lymphocytes are actually the T cells. Therefore, it is written like that most of the lymphocytes are actually taking part in adaptive immune responses, not in innate immune responses. All right. I hope this is clear. And if not, please write your queries in the chat box. And if you do not have queries, then we can go ahead. Okay. So there are differences in the innate immune response and an adaptive immune response. Mainly adaptive immune response is due to B and T lymphocytes. And in the last class, we also covered this antigen presenting cells, which is one of the antigen presenting cells. So there are three major type of cells which are antigen presenting cells. These, please mute yourself. Who is making this noise? I do not know actually. So, Kwashid, please mute yourself. All right. So, in the last class, we have discussed about antigen presenting cell. तो antigen presenting cells भाई होती क्या है? तो ये तीन तरह की cells होती थी. Last class में भी हमने discuss करा है. These are actually, so एक तो देखो यहाँ पे B cells हैं. ये एंटीजन प्रेजेंटिंग सेल की तरह से भी वर्क करती हैं. सो बी सेल मैक्रोफाजेस, right? And third one is actually the very important that are called dendritic cells. So these three major type of cells are actually called antigen presenting cells. Now see this, the B cells are already placed in this category called adaptive immune response. So B cells, which are actually antigen secreting, antibody secreting cells, are antigen presenting cells also. Macrophages are kind of phagocytes, which are actually part of innate immune response, but they are taking part in adaptive immune response also. And the dendritic cells, which are, the question has been asked that which of the following are antigen presenting cells, best antigen presenting cells. So the dendritic cells are called as best antigen presenting cells. So you can note this. If someone asks you this question, that which of the following is best antigen presenting cells, so always you need to put a mark over dendritic cells. I will tell you the properties why these are called best antigen presenting cells. But for now, you need to remember that the best antigen presenting cells are dendritic cells. All right. So what are the other differences? Specificity, if we talk about specificity, so the innate immune response cells, which are actually the phagocytes, neutrophils, eosinophils, and macrophages, these are non-specific to antigen. But adaptive immune responses, if they're able to show these, so they are antigen specific. Response time, actually these are part of innate immune response, and we know whenever there is an infection, जब भी कोई infection होता है या कोई भी foreign particle या pathogen encounter होता है, तो सबसे पहले जो work करता है हमारी body में system, उसको बोलते हैं innate immune system. इसलिए इसका जो response है वो rapid है. Generally कोई भी infection होता है उसके hours के अंदर या minutes में ही ये जो system है innate immune system, ये काम करने लग जाता है. So it will start working. So it is fast. But adaptive immune response is slow in terms of response time, because it will take few days to respond to any pathogen or antigen if there is an infection. All right. Generally, if you look at the diagram, it will generally take 7 to 10 days start working, right? But the innate immune system is very efficient in this way because it will take less time to activate, and response time is therefore fast. If we talk about diversity, so the diversity which is shown by this innate immune response, because the system is present since birth, it is having limited diversity. It will recognize limited number of pathogens. But adaptive immune response, because it is made by B and T cells, and these B and T cells are actually making 90 to 95% of these lymphocytes. Okay, so actually they are making 20 to 40% over and rough average 20 to 40% of all the white blood cells. Therefore, they are showing very high diversity over their surface receptors, right? So they are showing very high diversity. Memory response, which is actually shown by this adaptive immune response. So it is having this memory response, and this memory response question has been asked several times. So you need to know that the basic property, one of the basic properties or characteristics of the adaptive immune response is memory response. Major cell types we already have covered. So distribution found in the vertebrates and invertebrates, sabhi mein hota hai, but this adaptive immune response is found only in jawed vertebrates. So majority of higher animals are showing this adaptive immune response. All right. So these are some very basic differences between them, but you need to remember this, that memory response is shown by these adaptive immune response, and they are shown by the higher vertebrates. All right, so we move back to wait a minute. Okay, so lymphocytes, as we already have covered, B, T, and NK cells, they together constitute 20 to 40% of the circulating white blood cells and 99% of the cells in the lymph. So in the first class itself, we discussed about two types of circulation in our body. One is blood circulation, second is lymph circulation. So the lymph system is actually having these cells, major cell types which are present in the lymph, but in blood, in the WBC's category, these lymphocytes are making 20 to 40% of the white blood cells in the blood. Lymphocytes can be broadly subdivided into three major classes: B cells, T cells, and natural killer cells. Already told. Humans approximately having a trillion, that is 10 to power 12 number of lymphocytes. So these lymphocytes, which are B cells, T cells, and natural killer cells, are 10 to power 12 number of cells are present in our circulation in a normal human, circulating continuously throughout the blood and lymph, migrating into the tissue spaces and lymphoid organs. So in a human, you may have 10 to power 12 number of lymphocytes on an average. Now, how will you differentiate these cells? So they should have something common between them so that we can place them together under this class called lymphocytes, and they are having differences over their surface markers so that you can differentiate them like B cells, T cells, and natural killer cells. So this classification is depending upon the surface proteins which are expressed by the immune cells over their surfaces, which are known as CD markers or the cluster of designation markers. 1982 में ये सिस्टम आया था, इंटरनेशनल ग्रुप ऑफ इन्वेस्टिगेटर्स ने एक सिस्टम को अडॉप्ट करा जिसमें उन्होंने बोला कि जिन भी सेल्स के ऊपर कॉमन रिसेप्टर्स होंगे या कॉमन प्रोटीन्स एक्सप्रेस करेंगी उन सभी को एक ग्रुप में रखेंगे, और अगर वो अलग अलग शो कर रहे हैं, अलग अलग प्रॉपर्टी शो कर रहे हैं, then we will put them accordingly. So now we will discuss how this cluster of differentiation system is able to differentiate these B cells from the T cells and natural killer cells. So what is the specificity over B, T, and NK cells? Now look this particular diagram first, then I will show you one table where you will get this. So first of all, this is called common lymphoid progenitor cell, right? Which is actually coming from the hematopoietic stem cell. So please look into the diagram. So first of all, this common lymphoid progenitor cell will differentiate either into the natural killer cell, B cell, or T cell. Then I told you that this T cell further differentiate into the T helper cell and T cytotoxic cell. What is the difference between the two? T helper cells are showing CD marker 4 and TC cell are showing CD marker 8. So CD4, CD8. Then this T helper 1 population can be further subdivided into the groups depending upon the presence of some markers and their secretions, which kind of cytokines they will secrete, either into the T helper cell population 1, 2, 17, and T regulatory cells or T suppressor cells. So there are 4 different type of populations of these T helper cells itself. Now see this diagram. Okay, so there are T helper cells, there are T cytotoxic cells. These are only cartoons. Okay, so these are not actual cells, but throughout this cube you will see these designations, how they will represent the cells. Actually, the cell is something like this, which is shown in this diagram, but they shown this cell with the help of some surface markers. So always there is this kind of receptor. This is called T cell receptor because it is present over T cells. So either T helper cell or T cytotoxic cell. What is the difference between the two? They are both having this receptor called TCR, T cell receptor, but they are having differences. So you are able to see this T cell receptor, which is common in both, but they are having differences in the T helper cells and T cytotoxic cells because they are showing different surface markers. So if you see this blue colored receptor, which is actually CD4, right? So it is present over the T helper cells. And if you see this pink color receptor, this is called CD8. So it is called the T cytotoxic cell. So what is the difference between the T helper cell and T cytotoxic cell is a CD marker. So CD4, if CD4 is present, this is called T helper cell. If the CD8 is present, this is called cytotoxic cell. All right. Now, B cell is completely different because they do not have these T cell receptors. They are actually having B cell receptors. So these antibodies are actually called B cell receptor, BCR. So this is BCR, this is BCR. They are having so many BCR attached to the cell surface. What is the property of these BCRs? These BCRs are also called as antibodies, but they are membrane antibodies because they are already attached to the membrane. All right. So So they are having this portion which is called fragment antigen binding. So this is responsible for binding with the antigenic fragments or the complete antigen. So these are called fragment antigen binding. And if you are able to see, this FC region is already attached or integrated into the membrane of the B cell. So this is actually called the FC region or the fragment of crystallization. All right. So actually, antibodies are itself called as B cell receptors, whereas the T cell receptors are different. Now, if this B cell is activated by binding of an antigen, suppose an antigen will come and it will bind to the surface of this molecule. Suppose this is an antigen. So this will bind to the antibody molecule where it is having the site, specific site for its binding, right? So it will bind with it, and this cell will differentiate into a plasma cell. So this B cell will differentiate to make this plasma cell, which is a large cell. Now see the differences. Here you are having attached immunoglobulin molecules over its surface. यहाँ पे अगर आप देखोगे तो सरफेस पे बहुत सारे एंटीबॉडी मॉलिक्यूल्स और बी सेल रिसेप्टर्स प्रेजेंट हैं, लेकिन जब ये सेल डिफरेंशिएट होकर प्लाज्मा सेल बनाती है तो सरफेस से पे जो एंटीबॉडी मॉलिक्यूल्स प्रेजेंट थे, बी सेल रिसेप्टर्स प्रेजेंट थे, वो नहीं होते हैं, and it will differentiate, it will enlarge in size because it will have rough endoplasmic reticulum and Golgi body present inside the cell cytoplasm. Now what will happen, it will have genes for some proteins called antibodies. These genes will be transcribed and translated by the rough endoplasmic reticulum. Once rough endoplasmic reticulum will encode a protein, it will be glycosylated over Golgi body, and finally it will make an antibody molecule which will be secreted out of this cell. So it will make this molecule again, right? So this is called an antibody molecule which will be secreted out of the cell. So if you are able to understand this, so this is an antibody molecule, a Y-shaped molecule, which is actually having this hinge region where it is having this disulfide bonds, and heavy chain and light chain, which are together make an antibody molecule. So if someone asks you what are antibodies, so antibodies are actually glycoproteins, as these proteins are coming into the Golgi body and they are glycosylating these antibodies. Finally, they are secreted. So what is the difference between a B cell and a plasma cell? B cells are called naive cells. Naive cells means they cannot secrete antibodies. The antibodies are bound to their surfaces. But if they differentiate into a plasma cell, they will start secreting the antibody molecules at a pace around 1,000 antibodies per second. So this is huge number, right? So these cells can secrete up to 1,000 or around 1,000 antibodies per second. So each second they are secreting some 1,000 molecules of antibodies. So they are very efficiently secreting the antibodies, right? So they will make this humoral immune response. Humoral means fluid immune response, or the factors which are secreted into the blood. But we are actually today we are discussing this natural killer cells. So if you see the natural killer cell, actually they are under the microscope you are able to see these kind of cells, but they do not have any surface markers or surface receptors. Therefore, they are part of innate immune response. But if you are able to see into the cytoplasm, they are actually having these vesicles, which are actually filled with very potent enzymes, which can kill the infected cells or some of the pathogens, right? So they are able to kill, therefore they are called natural killer cells. So generally they are killing the altered self cells, which are actually called the tumor cells or cancer cells, which can proliferate unregulated, which are actually showing unregulated proliferation, and they are multiplying at an uncontrolled pace. So these cells, natural killer cells, will kill those particular cells. All right. So then we will move to page number 38. Okay. So everything is written in the text. If you want to read this, you can read actually very efficiently. They have explained everything. So also you need to remember this. Suppose there is an B cell as shown in this particular diagram, right? This is a B cell. Now, if this B cell will divide and it will make a clone. Clone is same type of cells. So the receptor molecules which are present over its surface, they are having same specificity. If there is a B cell, suppose I'm giving this a number 1, and we are having some other B cell, suppose number 2. So the antibody molecules which are actually secreted or which are present over its surface, they all have same specificity if they are part of the same population or clone. But if they are both are different, it is making a clone and it is making a second clone. So the receptors are different. One cell is having all the receptors of same specificity, whereas this cell will have receptors of different specificity from the first one. So you need to remember this. If the cell is actually coming from a clone population, same clone is called, because they are having different receptors over its surface. And all the receptors are actually showing the same specificity for any antigen. Therefore, they are called clone, right? So this one cell is different from the second cell if they are part of different clones or different populations. To understand this, let's discuss some theory. So the resulting population of the lymphocyte, all arising from the same founding lymphocyte, is actually called a clone. Clone is same type of cells which are actually showing the same specificity of receptors. Second thing is there are three different types of cells. One is called a naive cells. Naive is any cell or lymphocyte which is actually not encountered with any antigen. So mature B cells and T cells are ready to encounter the antigen, but they are not encountered yet. So they are considered as naive cell. All right. अभी तक अगर कोई एंटीजेनिक चैलेंज इन्होंने फेस नहीं करा है तो मैच्योर बी और टी सेल्स को बोलते हैं नेव सेल्स until they do so. So contact with an antigen induces naive lymphocyte to proliferate and differentiate. Actually होगा क्या अगर ये किसी एंटीजन के कांटेक्ट में आती है तो इसको प्रोलाइफरेशन होने लगेगा यानी क्लोनल सिलेक्शन होगा और ये डिफरेंशिएट कर जाएगी. डिफरेंशिएट इन द सेंस the B cell will make plasma cells and the T cell will make cytotoxic T lymphocytes, CTLs. So these will differentiate into both effector cells and memory cells. So there are two types of cells which will be generated. One is called effector cells, one is called memory cells. To understand this, let's go to page number 11 first. So suppose this is an antigenic challenge, some kind of foreign particles, either bacteria, virus, or protein. If it is given to some host or an vertebrate animal, so we are actually vertebrates, right? So mice is also vertebrates. So we are higher animals. Actually, we are having this adaptive immune response. So what you want to explain in this particular diagram, this mice or this vertebrate will show two kinds of responses. One is called humoral response, one is called cell mediated response. Humoral response because of B lymphocyte, cell mediated because of T lymphocyte. Now see this B cell, which is a naive cell, when it is coming in contact with this antigen or bacteria, it will differentiate and proliferate into a plasma cell and it will secrete large number of antibodies. As I told you, around 1,000 antibodies, but the number can vary from cell to cell. So around 100 or few hundreds antibodies up to 1,000 antibodies per second. So this is a huge number if we took this time factor into consideration. So per second actually. So this antigen elimination by these antibodies, because these antibodies will try to neutralize the antigen. It will bind with the receptors and they will try to remove this. Or they will opsonize these particles so that macrophages will come and will kill this bacteria. Either way possible. Second type of response is called cell-mediated response, which is due to the T lymphocytes. T lymphocyte, as we know, there are two types. One is T cytotoxic cells, one is T helper cells. So what will happen? T helper cells will be activated in this response due to antigen. They will bind with the, suppose this B cell, right? So B cell, as I told you, B cell will also work as antigen presenting cell. So this B cell, after binding this antigen to its receptor called B cell receptor BCR, they will represent this antigen to the T cells, or they will express some MSC molecules called MSC2 class 2 molecules over the surface. All the antigen presenting cells also have MSC class 2 molecules over the surface. They will express the peptide, right? Macrophages and dendritic cells can also perform this work. They can present antigen to the T helper cells. Anti helper cell, once recognizing the antigen, they will be activated and they will secrete large number of cytokine molecules. These cytokine molecules will have two functions. Either they will activate the B cells so that they can proliferate and differentiate, and they can also make some cytokines which will which will activate the T cells itself, right? So generally, interleukin 4 is responsible for activating the B cells. Interleukin 2 is responsible for inducing the T cells. Now, this naive T cells, when coming into the contact of antigen or induced by these cytokines, they will differentiate into cytotoxic T lymphocytes. So this cell is cytotoxic T lymphocyte, or CTL, which is able to kill the, this is called cytotoxic T lymphocyte, CTL, which is able to kill the tumor cells or altered self cells or the pathogenic cells from outside with by directly interacting with this particular cell. Generally, it is considered that if the antigen is extracellular, it will be killed by humoral immune response or by the antibodies which are secreted by plasma cells. If the antigen is intracellular, suppose it will able to infect some cell in the body and it is intracellular, it is present inside the cell, then that particular cell will be killed by the cell mediated response by the T cells, right? So these cytotoxic T lymphocytes, which are coming from these naive cells, after differentiation, they will interact with the infected cell and they can kill this. You can see this pathogen here. So this pathogen, if you noted this, they are coming from outside. They're pathogens, right? So this one is actually shown here. So if it is intracellular pathogen or virus, then it will be killed by the cell mediated immune response. Now back to the page number 38 so that I can tell you something else about this. Actually, we already discussed this. Two type of cells will be resulted. One is called effector cell and one is called memory cell. So three cells we have discussed: naive cells, effector cells, and memory cells. Effector cells carry out the specific function to combat the pathogen, while memory cells will persist in the host, and upon the re-challenge, which is actually called the secondary response, during the secondary response, the memory cell will take part and they will show this rapid response. Now, there is a table. Okay, so today we are more interested in natural killer cells, for example. So if you are able to see this complete table, and if I annotate this one, so please see this one, natural killer cells. Okay, so we are discussing natural killer cells, right? So natural killer cells are actually showing, so which are plus, so this is plus, this is plus, this is plus, means these are present. These CD markers are present over natural killer cells. Because natural killer cells are part of innate immune response, sometimes the question has been asked that which kind of surface receptors are present, which of the following are CD markers which are actually present over natural killer cells, because we know they do not have any receptors specific for any antigen, but they do have CD markers by which we can differentiate them from the B and T cells. But we know these are lymphocytes, right? all the cells which are written here, B cells, T helper, T cytotoxic, and natural killer cells, they're all lymphocytes. So they should have something in something common. And what is this common? Common is actually this one, CD5, which is actually present in all the cell types. So we can neglect this one. Actually, CD5 is present in all the lymphocytes. So they are called lymphocyte because they are having CD5. Not only CD5, they are also having 35. Sorry, which one was that? Oh, sorry, 45. So they also have CD45. All the cells, all the lymphocytes are also having CD45. So if someone asks you what is the common feature of all the lymphocytes, so they do have two type of CD molecules, cluster of differentiation molecule over the surfaces. One is CD5, one is CD45. Now, what is the differences from the B and T cells? How these natural killer cells are identified? So actually, they are having this, and if you see this CD2 molecule is also present over T cells, T lymphocytes. They are not present over B lymphocyte, but they are present over T lymphocyte. Therefore, we can also remove this one. This is not very important, but you need to remember this, that the CD2 molecules are present over T and T cells and natural killer cells outer lymphocyte, but they are absent from the B cells. Right? Now we are left with only two. One is CD56 and CD16. So which one of the following is major? So actually this one is more important, CD55. So this can be asked as a question, which of the following CD molecules is characteristic of a natural killer cell? So CD56, you will recognize a natural killer cell by CD56, because if you remember, we also have discussed CD16 molecules are also present over macrophages. In the last class, we discussed macrophages. Oh, actually, sorry, I forgot to share the video, but I will share the lecture soon in the group. Okay, so CD16 is actually also present over other type of cells, for example, phagocytes, like macrophages. Therefore, it is also not very much important, but they are having this feature which is very important called CD56 molecule, which is actually present only over natural killer cells. What else is important? So CD3 is important, for example, but this is not for natural killer cells. This is important because it is present. It is present in T cells. So all the T cells are having CD3, which is actually also called as T cell receptor. So if you remember the TCR, B cells are having B cell receptor, BCR, but these are having T cell receptors, right? So CD3 molecules is common to all the T cells. CD4, it is present in one population of the T cells, which is actually called T helper cells. CD8, other cell type, which is called T cytotoxic cells, are actually having the CD8 molecule. CD16, I already discussed. 19 and 21, they are actually present in the B cells. So actually 19 is called as CD21 co-receptor, right? So it is a co-receptor to the second one, and the CD21 is actually called the complement receptor 2, and it is also the same receptor is used by the Epstein-Barr virus. So this is also very much important. You need to remember this one also, that actually the Epstein-Barr virus, which is a kind of pathogen, is using the same receptor called CR2 or complementary receptor 2 or CD21 to cause infection in the B cells, right? CD28, which is also a type of receptor present in the T cells. So here you go. CD28, 32 present in the B cells, 35 and 40 all are present in the B cells only, right? So if you make a summary of this table, you can note, because this is so much confusing now. So for simplicity, what will you do? You will make a simple representation, right? So here I'm trying to memorize all these. So natural killer cells, only you need to remember CD56 56. So this is very important. And sometimes it may have CD16 also, but this is not very significant. So you can remove this CD16, right? CD56 is very, very important. Second thing is, if we are talking in terms of B cells, so B cells are having CD19, 21, and 21, 20, 32, 35, and 40. Right, so all these are present in the B cells. 32, 35, 40. Right, so also you need to remember that 21 is used by Epstein-Barr virus. So this is also very, very much important, right? So 21 is actually used by the Epstein-Barr virus, EBV. You can write EBV here so that you can remember this is using this to recognize or to do infection. All right. Oh no, it is not visible actually. And so sorry, actually, this color is not very much visible. So CD, first of all, we discussed that 19, 21, 32, 35, and 40. All right. So these are present in the B cells. So you can also write B cell here so that you can. Then there are third type of cells called T lymphocytes. So the T cells are actually having CD3, which is called T cell receptor, TCR. They also do have CD4, CD8, but they are specific populations will have CD4 for T helper cells and CD8 for T cytotoxic cells, right? So CD8 actually. Also, they do have 28, right? So I missed the 28. So CD3 and 28. Why this is not working? I do not know. 28. All right. So they are having T cells are having CD3 and 28. T cell receptor. So CD3 is actually called T cell receptor. They are also having CD4, but actually it is present over T helper cells and CD8 over T cytotoxic cells. So this much of information is given in this particular table. So you can remember them. Okay. So students, if you have any query, please ask.